I'm Jocelyn Compton, and I'll be bringing you the high-yield facts of epidemiology and biostats. First and foremost, as a strategy point, I recommend that you review this lecture when you begin studying to grasp the necessary concepts to understand subsequent lectures, and then dig into the nitty-gritty equations closer to your test date. These equations are high-yield facts that are best memorized for test day. They fade pretty quickly, and pretty easy to get confused. Consider writing them on your scrap paper as soon as you sit down at your test station on test day. You will most certainly get a handful of these plug-and-chug questions, and you will need to use the right equation in the right situation. Step 1 loves nothing more than to put answer choices down that reflect the right answers to the wrong questions. Common study designs tend to be highly board testable. The boards will likely describe a study and then ask what type of study was performed. The answers will look something like this: A through E offering several different study types as answers. Let's go through each of them. A cross-sectional study is purely observational and asks what is happening at this exact point in time, because it takes place in the present time. It collects data from a group of people to assess the frequency of disease and related risk factors, all at a particular point in time. It is a snapshot study design. In other words, it gathers data about exposures and outcomes simultaneously. While it can show risk factor association with disease, it does not establish causality. The characteristic measure for a cross-sectional study is disease prevalence. Do you remember what prevalence means? Disease prevalence is defined as the proportion of people in a population who have a disease at a particular time. An example of a cross-sectional study will be looking at smokers versus non-smokers and their frequency of peripheral vascular disease. Here, you're collecting data in the present moment to assess the frequency of peripheral vascular disease within a group of smokers, and you compare it to a group of non-smokers. Okay, so what's the major weakness of this type of study? It only shows correlation, but doesn't prove causation. Why doesn't it show causation? Because there could be another factor that causes both the disease and the risk factor. A case control study asks, what happened? Because it is observational and retrospective. It looks into the past and compares a group of people with disease to a group without disease. The cases are the subjects with disease, and the controls are the cases without disease. And in a case control study, you are analyzing previous exposure. The typical metric for case control studies is the odds ratio. In this context, the odds ratio is defined as the ratio of the odds of exposure in the case group to the odds of exposure in the control group. But how do we define the odds event? The odds of an event is the probability of the event occurring, or P, divided by the probability of the event not occurring, or 1 minus P. Let's define P1 as the probability of exposure in the context of disease, and P2 as the probability of exposure in the context of no disease. Then, the odds ratio is defined as P1 over 1 minus P1 divided by P2 over 1 minus P2. An example of a case control study will be looking at exposure to asbestos, which is a material found at construction sites and shipyards, and the development of mesothelioma. Here, you are looking into the past and comparing cases with mesothelioma to controls without mesothelioma, and analyzing their previous exposure to asbestos to see if cases with cancer had higher exposure rates. So let's suppose that the study finds that the probability of exposure to asbestos in patients with mesothelioma is 0.75, while the probability of exposure to asbestos in disease-free patients is 0.25. What's the odds ratio? Plugging the numbers into our odds ratio equation, we get an odds ratio that comes out to 9. A patient who was exposed to asbestos was 9 times more likely to develop mesothelioma. Next up is the cohort study. A cohort study can be prospective, in which case it asks who will develop this disease. Or it can be retrospective, in which case it asks who had exposure. It compares a group with a given risk factor to a group without the risk factor to assess whether or not the risk factor increases the likelihood of the disease. The cohort is the group with the given risk factor, and in a prospective study, you follow the cohort into the future to see if they get the disease of interest. The characteristic metric for cohort studies is relative risk, which is defined as the ratio of the probability of the event occurring in the exposed group versus a non-exposed group. The study answers the question, how much more likely is the patient to develop, say, mesothelioma if they work at a construction site with asbestos? A different example of a cohort study is the famous Framingham Heart Study. One thing this cohort study examines is uncontrolled hypercholesterolemia and future risk of myocardial infarction. Here, you are looking into the future to see if groups with uncontrolled hypercholesterolemia have a higher likelihood in the future of developing myocardial infarction than groups without uncontrolled hypercholesterolemia. Suppose that P1 is 0.75 and P2 is 0.25. Substituting these numbers into the relative risk equation, we find a relative risk of 3. Here's another way to compare case control studies and cohort studies. This point right here is the present. In case control studies, there are two groups at the present time, one group that has the disease and the other that doesn't. This study looks to the past to find the exposure. What would a cohort study timeline look like? Well, if we use the same present day, then you still have two groups. All the patients are actually healthy at this point in time, but one group has an exposure while the other was not exposed. The study looks into the future to see who is going to develop the disease. The last two types of studies are twin concordance studies and adoption studies. Twin concordance studies compare the frequency with which both monozygotic twins and dizygotic twins develop a disease. This measures the heritability of a disease. Specifically, the more concordance a disease shows for monozygotic twins relative to dizygotic twins, the greater the heritability of the disease. Adoption studies, on the other hand, compare siblings raised by biological versus adoptive parents to separate heritability from the influence of environmental factors on disease. Here's a summary slide of the types of trials for your reference. Let's do a boards type question together to see if you understand the differences between study designs. A study was conducted to determine the cardiotoxicity of azithromycin, a macrolide antibiotic that you learned about in the microbiology chapter. One group of patients taking azithromycin is compared to a matched group of patients not taking any antibiotics, and it was shown that azithromycin group had an increased risk of cardiovascular death. What type of study design is this? The answer is B, a cohort study, and it is prospective. We know that it is a cohort study because the question tells us we are comparing two similar matched groups. Additionally, one group of patients experienced an exposure, the azithromycin, and now are being observed for development of a disease. This is prospective because both groups are being monitored for future events. For the bonus point, what kind of information can be extracted from this type of study? That's right, relative risk of azithromycin exposure and cardiovascular death. Our next topic is the clinical trial. Clinical trials are experimental studies involving humans. They compare the therapeutic benefits of two or more treatments, or of treatments versus placebo. So, what are the characteristics of the highest quality clinical trials? They would be randomized, controlled, double-blinded trials, in which neither the patient nor the investigator know whether the patient is in the treatment or control group. How can a study be blinded? It can be single, double, or triple-blinded. When the study is single-blinded, the patient doesn't know if the pill they got was the real drug or the placebo, but the doctor and the researcher both know what the pill contains. In a double blind, neither the patient nor the doctor knows if the patient got the placebo or the real drug. In a triple blind study, not even the researchers analyzing the data know which pill the patient got until after the analysis is complete. More blinding results in less bias of patients, doctors, and the researchers. There are 4 types of clinical trials. Phase 1 through Phase 4. Each one progresses to the next. However, there are also new Phase 0 trials. These are called first-in-human or microdosing studies, where 10 to 15 healthy volunteers receive single subtherapeutic doses of the study drug to look at pharmacodynamics and kinetics and to confirm in vitro and animal data. Because they are getting lower than normal drug doses, you don't get any information on safety or efficacy. In contrast, classic phase 1 clinical trials study a new drug in 20 to 100 participants, usually healthy volunteers, to assess the safety, toxicity, and pharmacokinetics and pharmacodynamics of the treatment. Phase 2 clinical trials study a small number of patients, usually on the order of hundreds of people. These people have the disease of interest, and the point is to assess treatment efficacy, optimal dosing, and adverse effects. In pharmacology, what are the parameters used to describe efficacy, optimal dosing, and adverse effects? Efficacy is measured in terms of Vmax. Optimal dosing is measured in terms of potency, clearance, volume of distribution, and bioavailability, and adverse effects are measured in terms of therapeutic index. Phase 3 clinical trials study a large number of patients, often in the thousands depending on disease prevalence. Patients are randomly assigned to either the treatment under investigation, to the best available treatment, or placebo if there is no approved treatment. These studies compare the new treatment to the current standard of care. It's at this stage that the treatment may or may not receive FDA approval. And finally, Phase 4 clinical trials are designed as post-marketing surveillance trials of patients after drug approval. It's an effort to detect rare or long-term adverse effects. Take a look at this simplified chart of the 4 types of trials to help you remember. Phase 1 asks, is it safe? Phase 2 asks, does it work? Phase 3 asks, is it as good or better? And Phase 4 asks, can it stay? Let's do a flash quiz. What does it mean if a trial is single-blinded? Only the patient is unaware of the treatment being administered. The doctors and researchers know if the patient has received the standard of care, placebo, or the experimental treatment.